Great. So it's really good that then we're going to move up into the from cells and single molecules then into tissue microenvironment. And this is Richard Goodwin from AstraZeneca, who's also a professor at the University of Glasgow.
Thanks very much. So today I'll be talking about how we're using integrated molecular imaging technologies to offer unprecedented insights into the molecular microenvironment in our tissues, be they patient biopsies or any of our preclinical models. So I work within AstraZeneca and part of the business called Clinical Pharmacology and Safety Sciences. And so we're using these kind of techniques to understand both the efficacy and safety of our medicines in development. So over the next 18 or so minutes, I'll talk about how imaging and the convergence with AI and big data is really changing the way we can use imaging in drug discovery. I'll go into more detail around one of the techniques we've been using quite heavily, which is mass spec imaging. And then I'll touch on, throughout the presentation, the challenges and opportunities that we still need to address and overcome. So there really has been a convergence over the last few years that means that imaging is really letting us change information into knowledge. So no longer are we just collecting data that we're analyzing in one dimension. We're able to take that new data sets and integrate them and offer unprecedented insights. This is a convergence of the types of molecular imaging techniques we've heard about today that give us from an angstrom resolution to a patient resolution and everything in between, techniques and technologies that let us look for specific targets with great specificity or sensitivity, or look broadly at a number of targets simultaneously. At the same time, in the preclinical and clinical setting, there's a number of in vivo imaging technologies that have been developed and refined, which are giving us the ability to track and monitor without having to take out biopsies or take down animals to be able to look at a deeper molecular profile. And as I say, bringing all of this together, what's really let us start bridging between imaging modalities is the power of artificial intelligence. So as we've applied machine learning to understand our digital pathology and be able to speed up the analysis and reproducibility of our image analysis for our pathologists, the next layer is what can we see in the data that our pathologists and bioscientists can't interpret or can't image. And that's where we need to be able to use advanced image analysis to segment and process and simplify the data and look for those patterns and then display that back out in a way that our scientists can use that data. So the kind of molecular imaging techniques we have there's a whole range of spatial imaging techniques. So these are the ones people are more commonly aware of. So things like H&E, so our histology, looking at the morphology of our tissue, where our pathologists can see molecular events being recapitulated in the shape or distribution of cells within the tissue. So a very established technique, the gold standard in a lot of areas, but using technologies developed hundreds of years ago. The next layer up is something like immunohistochemistry, where we can use a probe or a target to specifically pull out the distribution of a specific target in a tissue. Or we can use something like a passive MRI to look at the morphology within a tissue, or we can use a probe-based approach in vivo using something like PET imaging. The next major shift we've been able to bring for imaging is the ability to multiplex some of these targets. So looking in one tissue at the same time, no longer just looking for the distribution of one target in that tissue, but looking for multiple. And this has let us start doing things like look at the effect of a specific compound in a tissue. So no longer are we using something like autoradiography to look at the distribution of our compound across an animal. We can look at the distribution of our compound and how that's affecting the tissue around it. Multiplex techniques include things like the mass spec-based techniques. I'll go into more detail, but also things like multiplex immunofluorescence, where you can use different detectors to detect multiple tagged targets. And the real game changer for us is when we start bringing all of this together. So the multimodal integration approach, where we can take our deep molecular tissue imaging with our in vivo imaging and our big data to give a holistic view of where our drugs are going to understand their effect on the tissue. But we can also use exactly the same techniques to look at the complexity of events occurring in our patients. So that gives us a better understanding of our next class of targets we want to start trying to develop new therapeutics for. And we're already using this across our project. So this is just showing that we can use the MRI to look at our whole body rat imaging. We can take out tissues and do deep molecular tissue imaging from mass spec or multiplex immunofluorescence or standard histology. And then all of that feeds into a advanced image analysis pipeline. And we can start seeing how we can make more quantitative measurements based on the tissue morphology. So no longer we were relying on a pathologist scoring to give a grading, but we're now able to make absolute quantitative measurements in multiple targets simultaneously. And we'll go into a bit more detail on that later on. So as I say, the bulk of my talk I want to go into is about mass spec imaging. So this is a suite of technologies that let us spatially resolve directly label-free molecules from the surface of fresh frozen cryosection tissue sections. And this can be any tissue. It can be patient biopsies. It can be preclinical animals. It can be plants tissues. It could be anything you can get onto a glass slide. We then ionize from the surface using a range of different ionization techniques, which give us different specificities and sensitivities. Each position across that tissue, each ultimately becomes a pixel in our image when we've done our processing. We ionize all the molecules we can detect. They go into a range of mass analyzers. From the mass spectrum, we take all of the data, typically hundreds of gigabytes worth of data per tissue, depending on the resolution of the mass spec and the resolution of the image. But for any mass we detect, we can then quantify its distribution and map its relative abundance across the surface of that tissue, giving a distribution of that specific molecule. And that molecule might be a lipid, it may be a drug metabolite, but we can see its distribution. In the same experiment at the same time, we can then map the distribution of another molecule. And we can keep repeating that for all the molecules we can detect, which gives us a holistic view of all of our targets in our tissue. We can do this in different ionization modes, or we can do it in different polarities. So we can really bridge that entire molecular landscape and recapitulate that in one single data set. And then we can also relate that data to the underlying morphology in the tissue. Some of our techniques are non-destructive, so we can take the very same tissue section, do our mass spec imaging, and then do subsequent immunohistochemistry or H&E analysis. The range of mass analyzers and ionization sources is ever growing within the mass spec imaging field. We've used pretty much all of them. So we have everything from MALDI, so high throughput laser-based techniques. We're heavily using in AstraZeneca DESI, so absorption electrical ionization. It seems particularly well suited to detect a lot of the molecular classes we're struggling to see by other techniques. Spatial resolutions are down to about 20 microns with a DESI, 5 microns with the MALDI. We can go down to a subcellular and approaching the nanometer resolution using the various SIMS platforms, so SIMS, NanoSIMS, and the 3D OrbiSIMS that we collaborate with external partners to get that really high resolving power, lower throughput, less rapidly deployed to projects, but we're giving unprecedented spatial resolution for specific targets. And we can go the other way. We can start using mass spec in the operating theatre. So we collaborate with Zoltan Takat from Imperial College, who was developing the interoperative rapid evaporatory ionization mass spec. So the ion knife, as it cuts through the tissue, detects the molecules that are generated in the gas, and we relate that data to the tissue data to give that kind of connection between the analysis in the patient and that in ex vivo. So this gives us an ability to go from that subcellular spatial resolution to the whole tissue resolution through all of the various techniques. And we can also go from an elemental mass scale all the way through to a proteomic scale. So what we need to do now is just decide which technique and technology we want to use to address which specific question. Within AstraZeneca, we're using it across our entire portfolio. We use it heavily for our drug distribution. It's pretty much replaced our use on autoradiography for looking at whole body quantitation. We can detect almost any molecule we're interested in, but we get the extra information of seeing all of the metabolites of that compound and also any effect it's having on the tissue around it. We can use that then just for efficacy, so we can look at the pharmacodynamic change in response to a therapy. So we can use not just protein biomarkers for efficacy, we can use the actual molecular molecules changing in response to the therapy, but we can also then do that for a whole class of molecules. We're not just looking at a single downstream indicator of efficacy. We can see every molecule in a pathway being modulated. We use it quite heavily within the safety organization, so we can understand things that aren't meant to be happening. If we see an adverse reaction or an adverse morphology within our tissues, we don't have to just flag that as being a concern. We can now go in and probe the events that are occurring. We can see if it's a drug particle that's being crystallized in the tissue, or we can see if it's a constituent of the delivery particle or the inhalation formulation, and then use that to help refine our programs to make sure that the safety doesn't cause attrition later on. And the area we're really excited to be using for model characterization, because we can look at that patient population and see the disease and its complexity. We can recapitulate that back in our molecular models and then make sure that they're best representing our patients. There are lots of challenges we still need to overcome. Sensitivity and resolution is always one. People want high resolution. When I started this about 8, 9, 10 years ago, resolution was about 200 microns resolution based on the laser diameter size. We get those down to 1 micron, 5 microns. There's still inherent complications going smaller than that with some of the laser-based techniques, but we always want higher spatial resolution. Higher spatial resolution means more data generated. It means longer acquisition times, and it means less sensitivity because we're ionizing less of the molecules at any one time. But with the mass spec vendors, we are really pushing the capabilities, and we are getting down to those cellular and near subcellular resolutions. We in AstraZeneca obviously are challenging ourselves with even more advanced therapies. So we're no longer just relying on small molecules. We're going all the way up to everything beyond antibody drug conjugates to the large genomic approaches as well. So they offer new challenges for us. And as I've alluded to, every tissue generates thousands of images because every molecule is its own image. So the data we generate from these technologies is astronomical. So being able to interrogate that to get the most value from it, rather than just saying what is the distribution of my drug, to be able to understand that complexity means we have to come about this from a different approach. I'll quickly now just go through how we're using this for something in an independent biomarker assessment model. So this is a recently published Cas9 genetically engineered mouse model, where we can see that the heterogeneity of the tumor distribution in the mouse lung really would challenge us to understand the molecular events that are occurring. Each one of these tumors is its own individual event, so we want to be able to look at each lesion and see how it responds to therapy, so we can understand the underlying genetics, but also the microenvironment and the immune cell population within that. So we take all of these tissue sections from our mouse lung. We do our histology, so we can understand what it looks like across the tissue from a histopathological view, so the more established approach for grading these different types of tumors. But we can then look at our molecular distribution. So this is pulling out just a GSH, so a metabolite, which shows that each tumor has a different molecular profile. So it's no longer just defined by the morphology of the tissue, but it's also defined by the fingerprint of molecules we're detecting by the mass spectrometer. And then we can take all of that data, we can recapitulate it into 3D because these are 3D sections taken from the same animal. And we can also segment that data based on the molecular profile. So using k-means clustering segmentation, we can see which tumor regions are most similar and most different and how they respond, which is great because we can now probe the ex vivo tissue. But the real advantage is we can also overlay this with the in vivo imaging. So in this example, we've got the PET and MRI from our collaborators at the Beaton in Glasgow, where they can do both metabolomic and morphological imaging of our tissues, which we then take down those animals, do the histopathology, do the mass spec imaging, and do the data analysis, all from the same samples from the same plane within the animal. So giving us the ability to look and baseline our animals before they go on to study. So no longer are we just randomly assigning them to treatment or non-treatment groups. We can see the morphology and the distribution of the lesions in the animal before we put on the therapy, see how they respond non-invasively using something like MRI. And then when they do start responding or don't respond, we can take down animals, do the deep molecular profiling to see the real heterogeneity and the molecular event driving the different changes. We can go one step further because this is all about integrated imaging from the same tissue. After we've done our mass spec imaging, we can apply something like imaging mass cytometry. So this uses metal tagged antibodies, which again are detected by a mass spectrometer, a different one. So we run it in parallel. We do the label-free mass spec imaging, label up with our antibodies, then put into an ICP-MS, ablate all of the molecules and detect the heavy metals and look at their distributions, which gives it the ability to get to a plus 30-plex immunohistochemistry image from the very same tissue. We've got the small molecular distributions. And this means we can do our biomarker assessment, define regions, and then look at the deep molecular profile and the phenotypic changes within that tissue. And this lets us look at things like the tissue architecture. We can have immune cell deconvolution, and we can do phenotyping all from the same tissue, all at the same time in tissue where we've looked at the distribution of our drugs and the effect on metabolism within those samples. And we don't just have to do this on ex vivo biopsies or tissues. We can do this on things like 3D organoids. So this is a recently published paper we've got with our collaborators in Sheffield Hallam, where they've got aggregoids where they take small clumps of cells. We can embed those in specific media, cryosection them through 10 microns as we do for our normal tissues, analyze them by mass spec, repeat that process through the organoid, and then recapitulate that back in 3D and start looking at things like oxygen gradients and hypoxic regions within the tissue. Also, simultaneously pull out all of the information around the TCA cycle and look at that distribution and how that's changed by any of the therapies we're administering to these models. Make quantitative measurements across all of that. As I've said before, this gives us the relative abundance, but just through the use of an externally applied calibration standard, we can turn the absolute detected molecule into an absolute quantitation, which gives us the real ability to look at how these molecules and metabolites are changing in response to our diseases. And from the same tissue at the same time, we can then look at the imaging mass cytometry, which gives us the ability to really probe response based on therapy. So, as I've alluded to, one of the big challenges is how do we integrate this data? And first thing we have to do with all of our data is digitize it, and this is something we're investing quite heavily. Is we have to be able to make sure we have access to all of our digital versions of all of our imaging data. The integration, analysis, and exploration is when we take all of that digitized data and start bridging it between modalities. We have to fuse data sets, and then we have to be able to use massive data sets down to ones that can be applied for clustering and analysis. What's fortunate for us within the mass spec imaging community is that we're able to pick all of the approaches that have been applied to big data elsewhere. So be that standard mass spec or look at genomic data or any of the platforms that have had huge terabyte data challenges, we can take the approaches that they've developed and apply them to our imaging data. Which means we can have a whole range of data reduction, segmentation, registration, classification, and fusion. That means that we can start bridging between those modalities within mass spec data, but also then just using other imaging modalities as just another data source in that process. But it is a big challenge, and this is just to kind of display that kind of information. So this is a small, low resolution mass spec data collected from what's the spatial resolution? 30 micron resolution from across a rat brain hemisphere, and you can see it's already generating 100 gigabytes worth of data. If we drop from 30 to 50 microns, obviously that data size goes by 4. So you can see this is a real challenge, especially if we're starting to do things in 3 dimensions. The last minute or so, I'll just touch on how we're really superpowering our ability to develop these techniques. Obviously, we work internally on this when we work with the vendors of the software, but we've been really fortunate to be part of a Cancer Research UK Grand Challenge. So we're part of the Rosetta team, so led by Professor Joseph Bunch from Imperial College and the National Physical Laboratory, where we got $20 million to really bridge the molecular landscape in cancer. So we develop it for CRUK and for cancer, and then apply to absolutely any tissues we're interested in. And this is getting us from that multiscale view from our whole patient all the way down to the molecular events. And the next little video should show how we're starting to do this. We now have the pipeline and the tools to take our sections, to do our histology, to do our lower spatial resolution mass spec imaging, use that data to segment out tissues, which we're then interested in to target the higher resolution, slower, more expensive techniques, to do integration of other imaging modalities, where in the last image we're down to the nanometer resolution and the SIMS data. So no longer do we have to randomly select areas. We can use the data we're generating from previous studies on the same platforms to target the regions where we're going to do our deep molecular profiling. And with that, I'll conclude saying with our integrated imaging techniques, we really are building a Rosetta Stone. So we don't have to use any one technique to ask any one question, but we can probe a sample with as many techniques as we can, and then understand which of the higher throughput, less expensive techniques give us the same information. So really driving our biomarker selection strategy going forward. And I think that's me out of time. So I'll stop there and see if there's any questions.
Great, thanks very much, Richard. You're very much in time, exactly. So I'll cross over to Chris and see if there's any questions on the board.
Yeah. Okay. Well, we have a question. Can you see mutant versus wild type morphologies in the tumor microenvironment, and how does that then impact the potential for treatment of the tumor?
Yep, so that's what we can see. We can see everything. The classification and segmentation is the real power of the technique, because we can detect everything that's derived from the underlying genetics, but also the immune cell population in those cells. We really do get an unprecedented view of what's occurring in those animal models. When we're applying it to the same data approaches to our patient populations, we have different challenges. We can't take necessarily the same controlled snap frozen, fresh frozen tissue section. So we have to maybe move to a fallen fixed approach, and we're developing techniques again with Imperial College to be able to probe those patient cohorts in a different way that might not give us the same molecular direct information, but does still let us differentiate between different subclasses of the disease, because ultimately there's still morphology in the lipid profile or the metabolites that aren't lost in the fixation process that can still segment data, and that gives us a new insight into the kind of patient population.
Okay, so any more questions?
Yeah, so I mean, I'd like to ask a question in terms of, do you think you're going to be able to then, look at de novo disease models, or, is it really dependent upon all this data? You need to know what you're looking for first, rather than say comparing, what's going wrong in a model.
Yeah, so that's again the real power of this. So we've traditionally applied these things to the hypothesis-driven questions. So we know the therapy or the disease is likely to have a modified pathway, and we'll look for that distribution. Because of the breadth of mass spec techniques we can now apply, there's pretty much no molecule that we can't detect or deconvolute. So what we're now doing it is in a much more hypothesis-free approach, where we just take condition A versus condition B and then probe that for the molecular changes. Once that gives us a pathway to follow, we can then apply a different imaging technique or a different polarity or spatial resolution to get even more information to differentiate between the different states. But as we're able to look at things like from tissue proteomics as well, because we can do tryptic digestions, we can look at glycan expression as well. We can really see every specific target we're interested in, every class. It really does give us the ability to go in an untargeted approach to look at the fundamental drivers of either the disease or the therapy.
Okay, and we also have a question from Andrew, who's on the panel.
So I was wondering about the very high resolution imaging. To what extent can you see subcellular distribution of different molecules, and to what extent is there sort of a background level versus, in one part of the cell there'll be a concentration?
Yeah, so that's our big challenge, and it's something we within industry have kind of our cutoff is that 5 micron resolution. Below that, we think you've got to be asking a very specific question. So the approaches using the nanosims approaches take a little bit longer. They either often use isotope ratios to be able to differentiate it. It's not giving you the information about if a molecule, or specific examples of drugs, are protein bound or if they're free. So you have to be more refined in the question you're asking. It moves away from that kind of holistic view of what's going on. But for specific questions, if we are targeting or tagging with a detectable probe, it does give us extra information. So I think that the mass spec based techniques give us some information, but all the other techniques we've kind of heard about today, that's where we want to bridge. We want this to be a continuation from that angstrom resolution all the way up to that patient resolution, and then bridging between all of the different techniques and technologies. So as we add cryo-EM, or we add the similar and compatible sample processing methods, we want to be able to be in a situation where we can hand those samples between the different assays to keep on probing further down, and also all the way back up to give that holistic view.
Thank you.
Okay, that's great, Richard. Thanks very much, and thanks to all the speakers this morning.